Alright, so this lecture is on chromosomes and the chromosomal basis of inheritance, basically explaining how our genetic information is passed from cell to cell through cell division. Okay, here are the list of objectives. Make sure that at the end of the OV, you come back here, check them out. If you have any questions, you can contact your teacher. Okay, so when we look at the DNA, we know what it is made up of. It is made up of a string of nucleotides, and they are, paired together, so you have two sides. Okay, as you can see here, we see that, characteristic shape of the double helix. But that's a lot of DNA, and it can be very unorganized at times. And so what ends up happening is we find during the life of a cell, during its cell cycle, there are two different forms that the DNA will take on, two different shapes in a way. And that is chromatin versus chromosome. Chromatin is sort of the loose, uncoiled form. So that would be looking at this right here. It's just, it's so small and thin. It's like a big ball of thread, okay? Very messy, very unorganized. And actually, when we are in interphase, of the cell cycle, that's fine because nothing really major is going on at that point where we would need to organize the DNA. But as you leave interphase and start entering cell division, you want to make sure that each piece of DNA is going to go into the proper cell. So things need to be organized. And so what ends up happening is the chromosomes, they're going to start condensing. And so as you can see here, it's starting to wind itself together. And it's going to be using these things called histones, which I'm going to show you on the next page. And it's coiling itself up and forming supercoils, and ultimately then we will get this chromosome structure. So if that would be that phase there. Make that go away. There you go. Okay, so that would be the M phase. Okay, during cell division, we're going to see our DNA condense for the purpose of being separated during the division process. So how do chromosomes coil? Just, a brief thing here so you can understand it. It's not really necessary to know this terminology. But what's going to happen is when we look at a chromosome, we're going to see our piece of DNA here, right? And these are A, T, C's, and G's, things like that. And then these little gray balls, these are going to be histone proteins. And they're going to sort of grab onto the DNA and twist it and turn it in and coil it as tight as possible. And they form these little nuggets of coiled DNA, and then the coils form on top of coils, and things get more condensed and more packaged more tightly. Think about if your job was to take a really long piece of string and you had to fit it in, like a pill capsule, okay? What would you start by doing? Well, maybe you might start by trying to coil the string, right? If you took it in your hand and balled it all up and rolled it to try and get it as small as possible so it could fit inside of there. So the coiling is going to do that. And imagine if you had, 23 or 46 of these different pieces. To keep them organized, you would want to coil each one into a smaller piece so you had like 46 little piles of your string all over the place. That's the same idea with the chromosomes here. So let me just flip back to this picture, okay? So just focusing in on what we're talking about here. The DNA molecule itself is not changing. We're not doing anything to the structure of the nucleotides, to their sequence. All we're doing is we are twisting and coiling them so that as we prepare for cell division, they are organized into smaller, more condensed units of genetic information. Okay, so what is a chromosome? That's what we just went over there. When you look inside each arm and leg, you're going to see that we have a single piece of DNA here. It's all tightly wound up. And when you look at your DNA, it is made up of gene segments, and these are not made of denim. Okay, gene segments are parts of the DNA that code for one trait. Okay, so for example, in my DNA, I have a gene and it codes for my hair color. Okay, in my sequence of the code, that gave me a reddish tint to my hair. Okay, but if you don't have that, then you're still going to have a gene for hair color. It's just that your gene sequence is going to be different, and I'll explain that a little more in a future slide. So it's important to understand the relationship there. Chromosomes are large segments of DNA that are coiled and bound together in an organized fashion, but genes are small sections of DNA within each chromosome that code for a particular trait. So we're sort of getting a hierarchy here. Okay? In your cells, you actually have two different types of chromosomes, all right? So you are a human, and congratulations to you. You have 46 chromosomes, okay? 46 chromosomes. Where did you get them from? Well, 23 of them came from mom, and 23 of them came from dad. So what did they give you? What had ended up happening is the 23 kind of got together with the 23 here. You get 46 total, but there are 23 pairs. So there's similarities between the chromosomes that mom and dad have given you. And we're going to talk about homologous chromosomes in a second. But basically, you have autosomal chromosomes, and these would represent chromosomes numbered 1 through 22. To keep them organized, we like to number them for our purposes. And basically, an autosomal chromosome is going to be the same, whether it contains the same genes on it, whether you're male or female. There's no difference there. So little things like your hair, your eye color, your ear shape, the length of your pointer finger, those are all going to be found on your autosomes. But you have pair number 23 here, which is very special because instead of just getting one from mom and one from dad, there's a difference between the two of them. So instead of being autosomal, we're now going to see a difference. These are your sex chromosomes, and you can either get an X or you can get a Y. Okay? Since we get 2 of every chromosome that we have, right, to make a pair, then this determines your gender. So if you inherit an X from mom and an X from dad, then that is going to make you female. But if you get an X from your mom and a Y from your dad, then that is going to make you male. So if you look at it, okay, mom can only give an X, right? When her cells are dividing and she's splitting and making her DNA, it's like, okay, an X here or an X here. It's no, there's no guesswork there. You're going to get an X chromosome from your mom. But with your dad, an X or a Y, only boys have that Y chromosome. There's a 50-50 chance, okay? When his cells are forming that are going to, give rise to you, he can either get an X chromosome in there as his sex chromosome, or he can get a Y chromosome in there as his sex chromosome. So really, the dad is the partner that determines the sex of the new offspring. And these are going to code for some gender-specific traits. Some of them are really not related to your gender, but they're just found on this chromosome. Like, for example, hemophilia is found on the X chromosome. And hemophilia is a disease which the blood is not able to clot properly, and so you don't form scabs the proper way. So if you cut yourself really bad, it could mean the difference between life or death, and not just, put a bandage on it and wrap it. It'll heal after a while. Color blindness would be the inability to distinguish the difference between certain colors and things like that. But in addition to these traits, there are also the traits that make you male and make you female, which are found on each of the chromosomes. So during cell division, your DNA condenses into these chromosome forms. And so if we pop a cell open when it's in the middle of division, we know that the DNA has already been released from the nucleus, right? That happens in prophase. And so during metaphase, if we interrupt a cell, what we're going to do is we're going to be able to find these chromosomes, and we're going to be able to match them up with their pair, okay? as things are forming here. And what scientists can do is they organize something called a karyotype. So it's literally a photo of your DNA in chromosome form, and we use it to look for any problems or any mistakes in how your chromosomes have formed. Did you make an extra one by accident? Are you missing a piece of one? Are you missing a whole one? Things like that. So here is a picture of a normal human karyotype. That's the name of this chart or this diagram showing all of the different ones. Notice numbers 1 through 22, those chromosome pairs, those are your autosomes. So they're going to be the same regardless whether you're male or female. But your sex chromosome, pair number 23 here, this is going to differ. So here you'll see two X chromosomes if you are female and an X with a Y chromosome if you are male. And no offense, guys, the Y chromosome is pretty small, but that's because it doesn't really take a lot of genes in order to make you male. The X chromosome is essential for normal fetal development, so everybody must have an X chromosome. You cannot be born without one. As you can see, male, female, you at least have one. And then a few genes on here on the Y chromosome, when they turn on and they produce proteins, namely hormones, that is going to determine whether you're going to be male or female. So you might feel really different as far as gender goes, but on a genetic level, there's not a lot that's different between male and female. Oops, okay. So just reviewing parts of the chromosome again, you know, we have our chromosome, the chromatin has condensed during prophase, and we already made a copy of our DNA during S phase, right? So when it was in that chromatin form, we made a copy of it. Now those sections of DNA, they've condensed, and the copies of DNA have found each other, right? So we talked about this before. So now what we have in this chromosome is we have sister chromatids. So let me just highlight this here. So here we go. This is one sister chromatid. And this is another sister chromatid. That's what we're looking at here. And so this is a pair, these are sister chromatids, and they are identical pieces of chromosome. Their DNA code is genetically the same. Okay, the sequence of all the DNA here is the same as the sequence of all the DNA here. And they're held together by a protein complex that sort of wraps around them, and we call that the centromere. Okay, so when we take a closer look, that's an actual, there you go. That's an actual image, like a photograph, essentially. I think it's a 3D image of, I don't know what machine was used, a micrograph, I'm going to assume. You see what we have here is we have the formation of our sister chromatids. This side is identical to this side. They are genetically the same. And so they're going to line up during metaphase, and then in anaphase, they're going to split. So into this cell, you're getting the same code and pieces of DNA and genes that are going into this cell. Okay, eventually that's going to be the end result there. So, why do we need to talk about homologous chromosomes? Let me pull this up here a little bit more. Okay, there we go. Homologous chromosomes. All right. Let me label some things here. This is a sister chromatid pair. This is a different sister chromatid pair. Okay? So when you look at this, we have two pairs of sister chromatids, and they're sort of hanging out together. Why are they hanging out together? Because they are homologous. And what does that mean? It means that they share genes. Okay? So for example, I don't know if you remember way back when in 7th grade, you did Punnett squares, right? So let me just put a little Punnett square up here. And you said, oh, okay, I have, big A here, and I have little a, big A here maybe. And then you would fill in the Punnett square, and what you were doing is you were predicting all the possible combinations that the offspring could have if these were the genes that the parents had. Well, what's the difference between the big A and the little a? They are the types of genes, okay? So just pop down here real quick. What I'm talking about is a new term. They're called alleles. So they are the different versions of the same gene. So going back to my hair color example, I have a gene for hair color. You have a gene for hair color. But my alleles on that gene give me the sequence that determines that I have a red tint to my hair. And your alleles may determine that you have a very dark tone in your hair, or you're blonde, or whatever. So there can be multiple types. Okay, so for example, when you look at earlobe shape, okay, if you look real closely here, I'll zoom in. My earlobe, it doesn't hang low. It sort of attaches right to the side of my face. You see that? That's an attached earlobe. But there's another form of the same gene that produces unattached earlobes, and that causes them to sort of hang low more. They don't attach right to the side of your face. They dip a little bit. And we'll take a look at traits when we get to genetics. But for the sake of explaining here, these chromosomes kind of hang out together with each other because this chromosome has the recessive form, the recessive allele, which in this case would be like attached earlobes. So here is the gene sequence for attached earlobes, and since this is the exact copy of that, it also has the gene sequence for attached earlobes. But this pair has the gene sequence for unattached earlobes, and this is its copy. And it has a gene sequence for unattached earlobes. So why do we have both of these? Why do we have this one and this one? It seems like there would be a fight. Well, there is going to be a fight. Remember how I said you get half of your genetic information from mom and half of it from dad? Here's where it is. They gave you chromosome number 1 through 23. So let's say this is, chromosome number 7 from your mom, okay? This represents chromosome number 7 from your dad. So they have the same genes on them, but they might have different alleles depending on what your parents had in their own genetic makeup to give you. So we've already talked about this before. Humans have 23 pairs of chromosomes. You get 23 of those from your mom and 23 of those from your dad. So that makes 23 pairs. You see how a pair would be times 2? So 46 chromosomes total. They are paired, meaning they don't actually touch one another, but they're paired in that they are the same genes, but they can have different alleles. This is a really tough concept. We're going to spend a lot of time on this in class. So if you have questions right now, please write them down, ask them in class, and we will go over this. All right. So when we look at the karyotype, and this is an actual karyotype, we add little markers to make them fluoresce in different colors like this. What you're looking at, this is taking the copies out. Okay, so we're not looking at sister chromatids. We're looking at single chromosome number 7 from mom here, and single chromosome 7 from dad here. Well, actually, it's number 1. Sorry. So this is like single chromosome number 1 from mom, single chromosome number 1 from dad. And so when we look at all of these pairs together, that makes up your entire genetic code. So there's a homologous pair, which is not the same thing as a sister chromatid pair. Okay? So when your cells divide, they can form different cells, and those cells are different based on the number of chromosomes they hold and based on what function they're going to have. All right? So in mitosis, our end result is that we have, oops, did that get cut off? Yeah, it did. That's okay. I'll fill it in. In mitosis, the end result is that we take a cell that has all 46 chromosomes in it, we copy that number of chromosomes, and then we divide it so that we get 2 cells that have the same number, 46 and 46. So let me just draw this here real quick. So we start with 46, then we up that number to 92, right? Because during S phase of interphase, we copy all of the DNA. Then during mitosis, we divide them so that we have two cells that are each containing 46 chromosomes. And remember, 46 is the full set, okay? Mitosis produces diploid cells. Di meaning two, you have two of each chromosome type, okay? So you start with a diploid cell and you end with two identical diploid cells. That's what that term means. Mitosis is the process of making more diploid cells. Another cell type you can make is a haploid cell, and this is what's so special about meiosis. Now we're going to, and there's a whole nother lecture on meiosis, but I just wanted to introduce this here very quick to you. Now we start with a diploid cell. It starts with 46. We divide it up. Okay, we multiply our chromosomes. Those chromosomes we then split through one division. But then in meiosis, there's another division that occurs. And so what ends up happening, sorry, I'm writing through this here, 23 and 23. So what ends up happening in meiosis is we get 4 cells that form, and they have half of the genetic information of a diploid cell. So instead of having 2 sets of chromosomes, there is only 1 set of chromosomes. And in diploid cells, whereas this makes all of our body cells, or the other term you could use would be somatic, S-O-M-A-T-I-C, somatic cells. Meiosis occurs simply to make your sex cells, the sperm and the egg cells. So sperm for guys and egg cells for girls. So haploid cells are only formed during meiosis. Diploid cells are only formed during mitosis. What's interesting is both cell division processes start with a diploid cell. Okay? So start getting used to some of this terminology. As we get into meiosis, we will explain those concepts a little further. Okay? So here's what's happening. You see the egg cell here has 1 copy of this chromosome, and the sperm here has 1 copy of this chromosome. But when fertilization occurs, they are going to go together into the zygote that forms, and that's basically your first cell of a new life. And now they are homologous to one another, right? Here we have a homologous pair. So when we look at our chromosome numbers, like I was saying before, humans have the number 46, okay? Chimpanzees, they have number 48, it's pretty similar. But then you have this fern here, which, looks like this. That has 1,262 chromosomes. A dog has 78, horse 64, cat 32. So the number of chromosomes that you have is not significant. That's, it's the total amount of DNA, and it's been broken up into these sections. We just happen to have 46 of them. Okay? So when we look at the human chromosomes, again, to review some key points here, humans have 23 pairs of chromosomes. That makes us a total diploid number of 46. So our diploid number is 46. What would our haploid number be? It would be half of that amount, right? Haploid, half. So half of that amount would be 23. Okay? Females have 23 homologous pairs, because when you look at that XX for their sex chromosome pairing, they are still homologous, right? An X and a Y, they're going to contain the same, an X and an X, they're going to contain the same genes. So not only do we have the first 22 pairs that are homologous, but the last pair is also homologous. Males, however, they have those first 2 homologous pairs, right? These are the autosome. But then their X and their Y, they don't contain the same genes. And so because of this, we consider that an unmatched pair. So they are an X and a Y, and they are non-homologous to one another. So males only carry 1 copy of each gene that is on an X chromosome, but females have 2. And this is going to have an implication for how different traits can be inherited and how there will be differences in males versus females because of that. Okay? So that's the end of the video lecture. I think this one is it's difficult, okay? Honestly, it really is a difficult lecture because it ties in some things we've already learned about mitosis, but it's also referring to things we have yet to learn about genetics and inheritance with meiosis. And so you're sort of stuck in the middle here where we're reviewing some concepts, but also stretching your understanding as well. So please write down any questions that you have, and we will take the time to go over this in class. We've got a number of modeling activities planned and things like that. All right, if you have any questions, please contact your teacher.